I'm going to talk today about the benefits of harmonising genomics across testing animals, foods and humans. And I'm an end user of genomics. I'm not someone who's a lab scientist. My background is in epidemiology, which is the study of disease and its patterns and causes. And I've done that for about 30 years, but we've been using laboratory science over the years, and it's become increasingly more complicated. And so hopefully I'm going to tease some of that apart, but really my main role today, and the take-home message is, as an advocate for whole genome sequencing, it really is like focusing our vision. I'm short-sighted, and the old tests we used to use, like phage typing and serotyping, they're like not having glasses on. So, just let me get this. So today I'm going to be talking about the importance of food safety, obviously. Why are we all here? Whole genome sequencing, talking a little bit about what the technology is and how it can help us. The application of whole genome sequencing to food safety. And I'm going to give you 5 promises of whole genome sequencing. I'm going to take it from a foodborne disease investigation perspective, and I realise if you're talking about food safety, that's after the horse has bolted, but I think whole genome sequencing offers many new promises. And then I'll just have a few concluding thoughts. Why are we all here? We're here for food safety, and this picture reminds me that the global burden of disease is more than just what we see in high-income countries. Overall, there's an estimated 600 million cases of foodborne illness every year and around about 420,000 deaths. But the burden isn't shared equally, and this is a picture of a young child. In under 5s, they represent about 8% of the population, but in terms of the burden, they experience about 38% of the episodes of illness, so largely diarrhoea, but also other illnesses, and around about 30% of the deaths as well. So if we're to fix our food supply, we really should be thinking bigger than just high and middle income countries. The other reason food safety is important, obviously, is that, as we know, foodborne diseases are epidemic in nature, and this is an example of our most recent problem in Australia, which is the incursion of Salmonella enteritidis into the egg layer flocks. We've never had Salmonella enteritidis in our egg layer flocks. We've had lots of other Salmonella problems, mainly Typhimurium, but here we see an incursion. Where did it come from? How did it get there? Is it going to stay? They're all questions we have at the moment, and there's lots of investigations going on. The reason enteritidis is an important serotype is because it can infect people through the egg yolk. So the actual Salmonella goes through the ovaries of the chicken and into the internal contents of the egg, meaning you can't have your eggs runny. So there's lots of questions to be asked, and I think they're reasons we need tools to understand what's going on. Foodborne illness is going to keep occurring. In Australia, we estimate that there's 4.2 million cases of foodborne illness every year. We want to see those going down. It is actually one of the big disappointments of my career for the last 30 years that I've been working in this area, and the lines just keep going up. The graphs just keep going up, despite all of our best efforts. There are many food safety disruptions going on at the moment. So globalisation has been happening for the last 30 years, but it keeps happening, and this Salmonella enteritidis is an example of that. Animals, foods and pathogens move freely throughout the globe with deregulation. Increased numbers of meals out, food fads, chicken liver parfait with recommendations to have them with red blood running out of them. Blockchain traceability is improving things, new food products and processes. Data analytics is making our understanding of food safety improving massively, so I think there's some really positive things. And also microbial investigation and whole genome sequencing, which is what I'm talking to you today, and I'm sure other people in this session will explain in more detail as well. What is whole genome sequencing? I recognise there'll be people in the room who know it intimately, but there'll be people who don't. It's essentially working out the entire genetic structure of an organism. We can do it to any organism. It provides a complete DNA profile, and it generates large data sets which have changed the nature of laboratory science. So we now have to have complex bioinformatic analysis. More and more I'm seeing these computer scientists in the lab, and so engineers, physicists, people who love big data are now in the lab, and they're also bringing with it new visualizations and making it far more complicated than just getting laboratory results. But more importantly, we can relate harmful organisms or pathogens to the source. Infected patients to food and food to animal. Just in more simple terms, what whole genome sequencing is, if we have a culture of an organism, so it could be Salmonella or E. coli, we might prepare the DNA, break up the DNA, and then we'll put it into a sequencing machine. And then we produce these files, which are essentially what are called reads, and they have essentially reads of parts of the genome. We've broken up the DNA, and they vary in length, but you can see here they're just lists of numbers representing different bases in the DNA. And if we think about that, it's a bit like getting a stack of newspapers, putting them into a paper shredder, and then we later have to reassemble them. And that's what the bioinformatic analysis does. It's reassembling these reads that have been broken up. If we think about it, then we put it into a phylogenetic tree, and this is courtesy of Torsten Seaman, who's a bioinformatician in Melbourne. I don't know, hopefully you guys have all of these particular snacks. I'm sure you have the one on the right there, which is very different. And essentially this is just showing us a visual overview of the relationship between things based on compared features. So in this example, we've got mint things together, like the mint patty and the peppermint crisp. And we've got things with nuts that are similar, like picnic and Snickers. And so the further away, the further up the chain they appear. So that gives you an example of how we might think about the phylogenetic tree. We call it a phylogenetic tree, and the differences are usually based on single nucleotide polymorphisms or changes in the structure of the base pairs in the genome. So if we think about what are the benefits of combining sequence data from humans, animals and foods. So we've got here a sheep, we've got here some sprouts. It's on my do not eat list. I hope it's on yours. I shouldn't say that too loudly. And if we have an organism coming from a human as well, say we've cultured this organism, and they might be the same organism, they might be E. coli, which are common in all of those animals and foods and humans. The real benefits we get is we can then look at relatedness. How related are they? How different are they on that phylogenetic tree? Can we see any clustering? Are they actually so close that they may actually be related in time, place and person? Is there any shared antibiotic resistances? We can learn pretty much everything about this organism based on what we see from a whole genome sequence. Virulence characteristics, are they more likely to cause sequelae like hemolytic uremic syndrome? So just as an example where we've been combining data from foods, animals and humans, this is a study in Australia which we've been conducting. Nigel is a part of that study, and it's been really fantastic collaborating with him. It's very similar to a study being conducted in New Zealand funded through MPI, which is very much along the same lines. We have similar Campylobacter problems in Australia to New Zealand and for many countries in the industrialised world. In fact, Campylobacter is a huge issue for many countries globally, even low and middle income countries, just not well documented. So we've been collecting food, animal and human samples in 4 states or territories in Australia, and you can see them listed here. And in Victoria, we only had a limited number of samples collected, but the others we've collected a range of different samples. And what we're doing here is we're using these in attributing sources, trying to work out, sure, we can find Campylobacter in sheep, but does it really relate back to the humans? We're also conducting a case control study, which is where we're looking at people who've got the disease, Campylobacter, and those who do not have it, and comparing dietary histories in their incubation period, and we'll be combining this with the mathematical modelling. I'm just going to show you a few results from that. These are interim. We will be sequencing around about 1,400 Campylobacters, and at the moment we're up to about 1,200. And a lot of them are Campylobacter coli, which is another species, but this is Campylobacter jejuni. I've also added in some isolates from other jurisdictions as well, which we've sequenced. We've put through that shredder and reassembled them. And these are from a national snapshot, so we're looking to see what kind of genetic diversity is. And we expected that when we looked at, say, Northern Territory, which is so different from southern parts of Australia or northern Western Australia, we would see that they were different, that you could actually say those isolates cluster. But you can see from this is a minimum spanning tree based on differences of single nucleotide polymorphisms in the core genome. And you can see here there's this huge spread of isolates around here. And so over in this left-hand side is more different than the ones on the top. But you can see there's a huge spread right across. There's no clustering by place. And if we add in the humans, you can see here that they are just all over the place. We basically see this big mess of genomes across the whole country. Then when we add in animals, you can see again that we're getting people infected with Campylobacter in somewhere like Queensland, and they match with a chicken isolate from Victoria. So there's this huge spread of Campylobacters, and it's not at this point showing us as much focus as we might have liked, but it's still very illustrative. And then if we add in the dogs, we can see that they're all over the place as well. Focusing in on one of the more common sequence types, this is sequence type 50, which is a particular strain from MLST. This is our most common strain in Australia. It's not the most common in New Zealand, but you can see it's present in all states and territories, present in all sources, including dogs and chickens, and it's highly diverse. There's 942 different single nucleotide polymorphisms between the top one that's been isolated from Queensland and the bottom one that's been isolated from Queensland. And you can also see these kind of clusterings here. This is a cluster of cases with very few SNP differences, and you can see they've come from New South Wales, Western Australia, Northern Territory and the ACT. So we're going to be using all of this and doing mathematical modelling, and it will allow us to say what proportion of different infections are coming from different sources. And just because we don't see the clustering by geography that we thought we might, we may well see clustering when we actually look at the mathematical modelling. We will be able to identify common sources. And it is largely about foods moving all over Australia. So chicken meat, beef, lamb moves all over Australia in vast amounts, and the supply chains are moving in ways that we can't fathom if we're just looking on a state-by-state basis. So just 5 promises about whole genome sequencing. Firstly, earlier detection of problems. It allows for earlier detection, and if you're applying it in a food safety context in a factory, in production, processing and retail, this is the ideal situation. We do not really want to see problems that foodborne disease investigators get involved in. It does rely on regular testing and sequencing. People need to be having a pattern of testing and sequencing. You can't just go in and sequence stuff without having a baseline of what you have actually got happening in the food supply chain. And I think capturing data within a company alone is expensive. These are usually, we're talking $50 per isolate for culturing, and then an additional $100 to $200 per isolate for sequencing costs, depending on what kind of company you're going with. There are some which will do it cheaper. And then you also have the analysis cost as well. Sometimes the analysis can be quite complex. I also think that if it's in a single company, you're underutilising the potential of what you have. And really the ideal situation is sharing. I'm not necessarily advocating sharing with government, but I think that's the best situation, is there's early communication of problems. The second promise is smaller outbreaks, and this is just a hypothetical example of an outbreak. These represent cases and the days of onset of this outbreak. And if we think about the first case occurring here at day 1, the health department might detect it at day 9, and then they might have laboratory confirmation at day 19 or something, and then their response happens after the peak of the outbreak. That represents our opportunity for control in that situation. So it's right up the end, and the outbreak's often declining, the foods have been consumed, or that particular food is gone from the marketplace. If we are able to get in earlier and to identify outbreaks earlier, we have a much greater opportunity for control. And this is true particularly where we've got widespread outbreaks and cases are related to each other. And that's the ideal situation for whole genome sequencing, is to identify cases that are related, but they may be spread in time or place. The third promise is to identify non-outbreak causes. And it's true that most cases of foodborne illness are not outbreak related, or we don't know that they're outbreak related. So for Salmonella, for instance, the most outbreak prone of all of the foodborne pathogens, maybe apart from norovirus, only 10 to 20% of cases are actually detected to be part of an outbreak. The remainder are the causes are undiscovered by health departments. It's challenging to identify the cause, and mathematical modelling offers great promise for identifying the sources of isolates. We can use machine learning because we've got huge amounts of data, and I think this offers opportunity for intervention. It's been routine in Scandinavian countries for decades. They have actually used mathematical models on phenotypic testing methods, and they've had engagement with industry and with government on agreements to actually share and communicate and identify sources. And that's why countries like Denmark have so few cases of salmonella in the food supply chain. Doesn't mean there's none, but it's lower. So the fourth promise is more specific attribution. And this is obviously really important from a food safety perspective. This offers the opportunity to identify specific foods in outbreaks and in source attribution. We know that when we do whole genome sequencing, and we have done trials where we've done all salmonella cases, we've sequenced them for 6 months in a given jurisdiction, we get more than 10% extra salmonella cases. And I think there's the potential for decreased impact to industry. There's obviously consultation required. This news clipping here is from an outbreak of Listeria last year, where it was actually a single farm, Rombola Farms in northern New South Wales, which was actually doing everything right, but the news about which farm it was came out fairly late in the piece, and there was industry-wide impacts, and I know that the rock melon industry really suffered as a result of that. Obviously, risk communication is critical, and both government and industry need to work in partnership on that. The fifth promise is a safer food supply globally. This picture of baby corn here is from an outbreak we investigated several years ago, and it's actually a Shigella infection. Shigella causes dysentery. And this baby corn, we had this outbreak of Shigella, which we weren't sure where it was coming from, and we never got to the bottom of it until we saw a report in Eurosurveillance, which is a public health bulletin in Europe, and the Danes had been investigating outbreaks of Shigella, and they linked it back to baby corn from Thailand. And so we communicated with them, and at the time, we didn't have whole genome sequencing. We had to share pulsed field gel electrophoresis images over the email. And that confirmed, yes, we did have that, but more importantly, we went back and interviewed everyone, and they'd all eaten this baby corn. The baby corn came back to the same packing shed in Thailand in the same week from a big outbreak in Denmark and outbreaks in Australia. But whole genome sequencing really ups the ante here. We've got global movement of food. Outbreaks occur commonly internationally from a range of commodities, and at the moment, health agencies are really not harmonising and utilising the potential here. It's disappointing to me that we saw more outbreaks that were internationally distributed when we were using pulsed field gel electrophoresis than we have been when we've been using whole genome sequencing. They're rarely detected or investigated. But WHO, the World Health Organization, is taking notice, and so is the Food and Agriculture Organization. And at the recent World Health Assembly, there was a edict given to invest in sharing of sequences and strains so that there could be far better detection of multi-country outbreaks. And that includes through InfoSan. And there are international databases where this kind of sharing occurs, including Genome Tracker, which is US FDA managed, and that has had successes in terms of identifying outbreaks crossing countries. So key challenges. Financial cost is obviously a big one, but we certainly know that from a public health surveillance perspective, typing human strains of organisms using whole genome sequencing is cost effective. Getting health, food and agriculture agreement can be challenging, as can involving industry. They have different aims and objectives, but ultimately it goes back to that picture of a child. We want to see a safer food supply globally. There are big practicalities. These are big data files. Storage and management is challenging. Analysis and visualisation is also challenging. We basically have to re-educate ourselves on how to do a lot of this. And then I think harmonising our surveillance approach. You don't have the same problems we do in Australia. We have difficulties getting our jurisdictions to agree. And that's just something we're used to. It's like herding cats. And also sharing sensitive data. We recognise that whole genome sequencing can be so specific it can pinpoint individual parts of a factory or individual product lines. So it just is a real game changer. Finally, I think this is a picture that we would like to think about, and the bottom aim or outcome is public health surveillance and disease control, so happier people. And we really do see whole genome sequencing at the middle of this. If we've got food and animal sampling, we've got isolates from humans, we can identify risk factors, we can look at geographic and seasonal variation. We can learn about antimicrobial resistance. I haven't really talked about that today, but from the whole genome sequencing, we can infer an organism's potential for resistance to different antibiotics, which is of global critical interest. And we can also analyse sources. And I think when we do source attribution using mathematical modelling, it actually takes some of the heat out of it. It's not done in the public spotlight of an outbreak. That's when we want to do it. So thanks very much.